are your theme 4 first set of video notes on genetics and heredity. So the focus of this video in particular is going to be mostly on just some intro Mendel things and just some intro vocabulary for this theme. So let's get started. So Gregor Mendel is the father of genetics. He was a monk and he spent his adult life studying how parent plants pass traits on to baby plants. And the thing that he was most famous for? Peas, of course. So why peas? There's lots of different reasons why he chose to study peas. The two major reasons are the fast reproduction and large quantities of offspring. Now there's 7 traits that peas have, which also made it a really important thing for him to study. So there's seed pea color, flower color, seed pod color, seed shape and texture, seed pod shape, stem length, and stem flower position. So lots of different traits to look at. When we talk about something called true breeding, and this is what Mendel was trying to do, is it's breeding for a specific trait. So to start his studies, he needed true bred plants. So to do this, he controlled pollination, or the fertilization of the plants, and after self-fertilization, these true bred plants would always produce the same trait. So another term you may be familiar with is the term purebred. So what is meant by purebred? Same thing, true bred, purebred, it's always going to produce the same trait when reproducing. For example, a poodle bred with a poodle is always going to have a poodle puppy. You're never going to have a beagle puppy because those are purebred species. So what if purebreds are mixed? So once Mendel got his purebreds, then he started working on crossing them. So he wanted to study the inheritance of pea plants in general, and he mixed his purebreds. And this is something called cross-pollination. So there's some important terminology on this slide that you need to be aware of. Definitely make sure you write these things down and kind of star them and highlight them. That way you know they're important terms. So the pea generation is the parent generation. The parent generation produces the F1 generation. And then the F2 generation is the offspring of F1. So if we look at this in context, F2 are the grandkids of the P generation. And you'll see this terminology when you start doing practice problems in class. There's also 2 terms, dominant trait and recessive trait. So if something's dominant, you're going to see it or it's expressed over another trait, usually the recessive trait. And the recessive trait is one that cannot be expressed over the dominant trait. So traits is a term I just used, but traits and alleles. So how are traits even carried in an organism? They're in the genes. So if you remember from the last theme, those genes are segments of DNA that code for a specific protein. The protein results in the trait. So in conclusion, one characteristic equals one trait. Then you might be thinking, okay, well, what are alleles? So alleles are alternative forms of a gene. So alleles are still genes, but they're just different forms of it. So for example, in Mendel's peas, there were 2 alleles for flower color, purple and white. Flower color itself is the trait, and then the alleles are the different varieties. And these are what's represented by letters when we do genetic crosses or Punnett squares. So as we take a look at Mendel's crosses, so he took purple purebred plants and crossed them with white purebred plants. Well, what do you think happened? They were all purple because purple was the dominant allele. Now, Mendel didn't necessarily know that purple was dominant and white was recessive, but this cross showed him that purple was definitely the dominant trait. So if you look here, the parent plants or P generation, purple is big A, crossed with white, which is little a. The offspring were all purple, but they're heterozygous, meaning they're big A, little a. So then if you take 2 of those offspring, so 2 from the P generation's offspring, which gives you your F1, that would be purple times purple, which are 2 heterozygotes. As a result, you're going to get those purple flowers, of course, but the white flower shows back up because you have recessive alleles. So he had 3 important conclusions. So first, the law of dominance. Simple enough. Some alleles are dominant, some alleles are recessive. Then he had the law of segregation. So this just means that 2 factors control each gene, and these factors are what separate during meiosis when the gametes are formed. So for example, short and tall alleles are going to separate into different gametes or sperm and egg cells during meiosis. They're not going to stick together. The final thing is the law of independent assortment. So this simply just means that different characteristics are independent of each other. So plant height, for example, is independent from flower color. This is just like something with humans saying, well, if you're tall, you're going to be skinny. Or if you're short, you're going to be fat. Those are separate things because you see short, skinny people and tall, overweight people, but they're not connected. So this is showing that independent assortment. Every different trait is independent. Now there are some other things with this, but we'll get into that later. Two other important terms are genotype and phenotype. So genotype is the type of allele, and there's three possible kinds. So you have homozygous dominant, heterozygous, and homozygous recessive, but in this class we're just going to say recessive. So you're either homozygous dominant or just dominant, heterozygous, or recessive. And you can look through there showing how that's represented. So dominant is 2 capital letters. Heterozygous, the prefix hetero meaning different, is 1 capital, 1 lower, and recessive is both lowercase. Phenotype is what the trait looks like, and the really easy way to remember this is phenotype equals physical. Write that down in your notes somewhere. Phenotype equals physical. Genotype equals gene. This is a really, really commonly mixed up terms. It's really important that you have an idea of how you're going to remember this. So let's practice a little bit here. So for the genotype listed below, give the phenotype. So black fur, or capital B in guinea pigs, is dominant to white fur, which is represented by a lowercase b. All you're going to do is list the phenotype. So pause it and write out your answers. All right, so you should have gotten capital B is black. Capital B, lowercase b is also black. And lowercase b is white. All right, once again, now you have the phenotype, so give the genotype. So pause it, take a minute, and fill that out. All right, so as you can see, there's 2 possible genotypes for the black toenails and only 1 genotype for the clear toenails. So clear is recessive, so that's going to be 2 lowercase letters. And then the black toenails are dominant, so you can have a dominant, so 2 capital letters, or heterozygous, which is capital and lower. Final thing we're going to discuss is Punnett Square. So why do we even do a Punnett Square? The whole point is to predict the possible offspring of a cross between 2 known genotypes, known being the key term there. We use those results then to predict the chance of a trait being expressed, and we will do lots and lots and lots of these in class, and you'll learn lots of different ways to test things out. So let's just take a look at what they are. So they're just 4 little boxes in a square. And all you do is you put the alleles of the parents on the top and the side. So for example, in this first one, big A, little a. So big A would go on top, little a to the side. And when you fill it out, you always fill it out sentence order. So capital should be before lowercase. Then we'll do another one here. So capital lower, big, big little, little. So hopefully that's a good review, and we will start doing practice problems in class, and you'll get lots more practice with this, like I'd said. And that's it for your notes.